have told water cell organelles. So these are the structures present inside the cell. They have got a particular function, a specific function to play in the cell. Okay, whether it can be the animal cell or the plant cell. So what is the structure? Cell organelle today we are going to discuss is about a vacuole. So this is one of the major difference between the plant cells and animal cells. Earlier also we have told the most important difference between the plant cell and animal cell is the presence of the cell wall. The outermost covering layer in the plant cell is the cell wall. The outermost covering layer in the animal cell is the cell membrane. Okay, so like that was one difference. One more difference is this one. Okay, regarding vacuole. So large size vacuoles are present in the plant cells. Okay, small size vacuoles are present only in microscopic animals like in amoeba and paramecium. And in general, if you say, so vacuoles are absent in animal cells. Okay, right, we'll see in detail about that. Now before we start with what is the function, how does it appear, where it is present. So let us try to understand this vacuole by doing one activity. So if you see the textbook, activity 5. Okay, so how to observe the vacuole in the plant. So for this selectively we have taken, so special type of the plant, that is desert plants. So what are desert plants? So plants which grow in the areas where there is low rainfall, less rainfall or scanty amount of rainfall. So they are called desert plants. So what type of number of desert plants you might have seen, you might have observed? Most commonly available in your house also one plant which is called as aloe vera. Okay, example for desert plants is one aloe vera. Then we have got cactus, CACTUS cactus. Then we have got opuntia, OPUNTIA opuntia. Then next one more we have got Cereus, CEREAUS Cereus. Okay, so like this we have got different type of the desert plants. So you can take this type of the desert plant material. So such a type of plants, so hold the water in their leaves or in the stem. So generally leaves are absent or reduced in the desert plants. So it is the stem which involves in the food preparation, photosynthesis. So we call it as a succulent stem. Okay, which holds the water, right? Such a type of the succulent leaf or stem we are taking a small material from any plant. So preferably the textbook is given a cactus plant. Okay, torch cactus we call it as. Why it is called as a torch cactus? It appears to be like that of the torch. So it is in that shape. That is why it is called as a torch cactus we call. So small portion of it, stem region, succulent stem we are taking. Okay, thin section of the stem we are taking and put it in a watch glass containing water. Now to this particular watch glass containing water, the material is ready there. Add a solution. What solution? Safranin solution. So earlier in the nucleus we have seen 1% methylene blue we have seen. Okay, these are the particular colors which are absorbed by the cell organelles. So methylene blue, 1% methylene blue was absorbed by the nucleus, which could be clearly seen under the microscope in the cell. Like that we have got here one solution we are taking, one color, one dye we are taking, which is called safranin solution. So 2 drops of safranin solution, put it into the watch glass containing the succulent thin section of the succulent stem. Okay, so wait for a few minutes, couple of minutes. Then now take that particular material after adding the safranin solution, put it onto the glass slide. You need glass slide. Okay, then once you have dropped the particular material over it, so cover it with the cover slip. Okay, cover it with the cover slip. Remove the extra water by using the blotting paper or a tissue paper. Okay, now start observing under the microscope. So already we have told there are 3 different type of the microscopes. Okay, of course at the school level or college level we will be using only 2 type of microscopes. Okay, so that is a simple microscope and compound microscope. So if you observe with a low magnification, so first observe this particular structure under the light microscope or simple microscope which is with low magnification, low power. Then again try to observe the same slide, okay, temporarily mounted slide under the high magnification, that is preferable in the compound microscope, under the compound microscope. Then what type of structures you find? Some large empty spaces you find. So these large empty spaces inside the cell are called as vacuoles. Are called as what? Vacuoles. So if you see, just observe in the under the microscope. Imagine this is the cell what we have seen under the microscope. Okay, in the plant cell already we have told large space, particularly in the matured plant cells. Maximum of the space is occupied by a large structure like this. So what is this? Nothing but this is nothing but the vacuole. Then where is the nucleus? The nucleus is pushed to one side in the plant cells after the formation of the large vacuole. How is this large vacuole formed? It is formed because of the union of several small vacuoles. So a number of small vacuoles combine to form a large vacuole, and in the matured cell you can see maximum space, okay, is occupied, and this large empty space, okay, structure is nothing but what we call vacuole. This is the nucleus. In the beginning, okay, when the cell is very young, the nucleus will be in the center, but it is a matured cell. You can see the nucleus is pushed to one side because of the formation of a large vacuole. Okay, and what are these containing? So these are fluid filled sac-like structures. They contain a liquid material, water-like material. It is called as what? Nothing but fluid, and it has got a particular name. What is that fluid? We call it as vacuolar sac. The fluid present inside the vacuole is called as vacuolar sac. Fluid inside the vacuole. Then what does this contain? So this fluid is nothing but the waste materials which are present inside the cell are collected and they are thrown out because of this large structure called as vacuole. And this vacuole is covered with a covering layer. It has got a covering layer. Okay, so like this. Earlier we have seen in case of nucleus is covered with one membrane. We have told it as nuclear membrane. Like that we have got vacuole is also covered with one membrane, outermost covering layer, which is called as a tonoplast. It is called as what? Tonoplast. Outer covering layer of the vacuole is called tonoplast. Okay. Now coming to regarding the size of the vacuole. So they are generally small in size. That is why we say in general vacuoles are absent in animal cells but are present only in microscopic organisms. What type of microscopic organisms? Suppose if you take like amoeba, okay, or if you take paramecium, okay, try to recall these examples. Already you have studied in 8th class or even in the beginning introduction first part we have seen. What do you mean by what is the shape of amoeba? What is the shape of paramecium? Amoeba is irregular shaped, paramecium is slipper shaped. It is called slipper animal and this is called as shapeless animal. Suppose like if you take amoeba, let us try to draw the amoeba diagram, a rough diagram. So like this. Now this is amoeba, irregular shape. So apart from nucleus, so this structure is the nucleus we have told. Apart from nucleus, what are the other structures present inside the amoeba body? You find two important structures. So this is one, okay, and this is one more. So what are these? So one is related to the food intake. It is called as the food vacuole. It is called food vacuole. So it is responsible for collection of the food and helps the digestion of the food, equivalent to digestive system in human body. Okay, so this is a primitive organism. Very small structures are present. Next, one more structure is present. What is this? It is called as the contractile vacuole. Contractile vacuole. So like this, two types of vacuoles are present in amoeba body. Same two types of structures. Same food vacuole, contractile vacuole are also present in paramecium. What is the function of the contractile vacuole? It collects the excess water entering into the animal. That can be amoeba or that can be paramecium. So the water, excess water entering into the body, animal body, and the waste substances are collected and thrown outside the body. So that is why the function of the vacuole. Okay, you can see here exports unwanted substances from the cell. So what do you mean by exports unwanted substances? Means throwing out from the cell. Okay, apart from that, it has got one more function: maintains the pressure inside the cell. Turgor pressure we call it as, and it occupies the maximum space. So particularly in the matured plant cell. Okay. Okay, so just now we have seen about the vacuole. So it is the one which maintains the Turgor pressure, occupies the maximum space in the matured plant cell, and it is meant for exporting the unwanted substances, okay, from the cell. So one of the most important structure, and it is a difference between the plant cell and animal cell. Presence of the cell wall, presence of the vacuole, and presence of the chloroplast. These are the three differences between plant cell and animal cell. Okay, now moving to one more cell organ, ribosomes. Yesterday we have seen minute small tiny particles scattered in the cytoplasm called as lysosomes. Same like that. Yesterday we have given one name to lysosomes, suicidal bags of the cell. Like that we have got same small granules like structures so present in the cytoplasm. What are these? So these are called as nothing but ribosomes. What do we call them as? Ribosomes. Okay, and they are formed of RNA proteins. And already we have told earlier also there are two types of genetic materials. When we have discussed about the nucleus, we have told nuclear material is present inside the nucleus, and that can be of two types: DNA, RNA. Okay, DNA means deoxyribose nucleic acid, RNA means ribose nucleic acid. Okay, right. Now coming to regarding the types. So based on the presence of the ribosomes, whether they are freely present, okay, here and there one individually present, or whether they are present as a group. Okay, suppose like 10 to 15 ribosomes present at one particular place, okay, attached to some structure, that is called one group. So based on the presence, so the ribosomes are differentiated into two types. We call it as free ribosomes, first type, and second type are called as grouped ribosomes. They are called as grouped ribosomes. A particular name is given to grouped ribosomes. So a group of ribosomes present in one particular place, they are called polysomes. What do you call them as? Polysomes. Poly means many, more in number. Okay, and free ribosomes, they can be present anywhere throughout the cell, inside the cell. They will be easily floating in the cytoplasm. Same like other structures like nucleus present floating in the cytoplasm. The ribosomes also will be present individually here and there. Okay, they are called free ribosomes, and second type it is called group ribosomes. Whatever it can be, whether it is free ribosomes or group ribosomes, what are they meant for? Their main function is, okay, they are useful for the protein synthesis. It is the place where exactly the protein synthesis takes place. Site for protein synthesis we call it as. And yesterday we have seen about ER. What is this ER? Endoplasmic reticulum. Okay, we have drawn the diagram also about the nucleus. From the nuclear membrane, you can see number of sac-like, tube-like structures are arising, which we call it as endoplasmic reticulum. And we have told this is of two types: SER, smooth endoplasmic reticulum, where there are no ribosomes attached, and second type is called RER, rough endoplasmic reticulum. Rough endoplasmic reticulum. Okay, so you can see here attached ribosomes. Where are they present? They are present attached to the surface of the rough endoplasmic reticulum. RER means rough endoplasmic reticulum. Okay, and useful for what? Protein synthesis. So this is all about the ribosomes. Of course, in your higher standards, you will be studying about still further differentiation of the ribosomes. Just little information I am giving here. So ribosomes are basically two types. One is called 70S type of ribosomes, and another is called as 80S type of the ribosomes. Again, these are further again differentiated. Okay, of course, that is all not there now at 9th standard. Intermediate level, you will be studying more about the ribosomes. On the whole, remember the function: protein synthesis.